Okay, can you see my screen?
Yes. Okay.
Okay. So thanks for giving me the opportunity to present my work today. I'm going to present you some last results that could be used to manage, to limit the fungicide use in the management of banana leaf spot disease. My name is Jean Carlier. I'm not sure. Okay, sorry. I'm not sure I took the right one. The leaf spot disease of banana are caused by 3 major fungi. From population genetic analysis, we found a higher level of genetic diversity in the pathogens in Southeast Asia. And so we suspect that the origin of this pathogen is from these regions, as it is for banana. The first described species was Pseudocercospora musae. It spread in the Pacific in the 1910s and in Africa and Latin America from the 1913. Pseudocercospora fijiensis, causing black zika or black leg disease, was described later and spread in the Pacific in the 1960s and in Africa and Latin America in the 70s. This species is more pathogenic than musae, and then it replaced musae in most places in Africa and Latin America. The last species, Pseudocercospora emusae, causing emusae leaf spot disease, was discovered later in 2000s. It's present in some part of Asia and has been described only in a few places outside Asia. So 3 species are originated in Southeast Asia, but the distribution of the 3 species in these regions is not well known. From population history, the population history of fijiensis and musae has been extensively studied, and from this study we showed that the intercontinental dispersal was mainly due to human activities, and the intracontinental dispersal was due mainly to natural pathogen dispersal, and in some cases to human activities. So black leaf streak disease is today the most important disease. So I'm going to focus my presentation on this disease. It causes this important defoliation, and this defoliation lead to lead loss following reduction of photosynthesis. But also because to be exported, the fruits are harvested green, and they are matured in the country where they are exported. The defoliation also causes a reduction of green life, and then an early ripening of the fruit, either directly in the field or during the shipping. So this fruit can't be sold, and it's lost. Fijiensis has 2 reproduction cycles, an asexual reproduction cycle with a reproduction of conalia, but also sexual or regular sexual reproduction cycle with reproduction recombination. And also this species has very large population size. The first sequence, the first genome sequenced showed that this species contain more than 50% of repetitive DNA, much more than other related species, as you can see here, in color other than blue. And so because of these characteristics, we suspect that fijiensis has a high evolutionary potential. To control, so as you may know, the cultivation of cultivar around the world, not in Asia, but in your part of the world, is based on the very susceptible group Cavendish. with no diversity. And so to control this disease in big plantation, a huge amount of fungicide is used until 60 kilograms per hectare per year. In some country, like in French, in West Indies, we use a warning system, which is useful to limit the number of application or treatment. But this system is difficult to apply in very humid regions. So we need new research, and new research has been conducted to improve agricultural practices and to create new resistant hybrids. For cultural practices, the effect of hedgerow on the spore dispersal, either Conidia or Ascospore, was tested experimentally in following such a design with hedgerow here, and we follow the spread of disease from sources and using a plant trap. And with this system, we found a significant effect of both Ascospore and Conidia dispersal of hedgerows. Also, we are studying agroforestry system. So in the first study, about 8 plots in Costa Rica was studied. And we found that the plant richness at the low stratum between 1.5 to 5 meters decrease lesion density as an effect on spore abundance and slow down disease evolution. So association with other cash crop like cocoa are being tested. But these first results suggest that interspecific diversification could be exploited. But further studies are needed to better understand the underlying mechanisms and to define how to do it in commercial plantation. But certainly, the new resistant hybrids will be the main means to control this disease. And the first resistant hybrids were created a few decades ago by the foundation, by a foundation in Honduras. And this hybrids has been cultivated at large scale in some Caribbean countries like Cuba and Dominican Republic. But unfortunately, after 5 to 10 years of cultivation of these resistant hybrids, we observe that the disease level on this hybrid has increased, and today it's more or less similar to the one observed on susceptible cultivars. So we suspect that pathogen population, because we know that the efficiencies could have a very high evolutionary potential, we suspect an adaptation of pathogen populations. To test this hypothesis, we collected samples from different locations in Cuba from 2 different types of plots. One of the plots were planted with resistance hybrids, FIA 18, which has a quantitative resistance, and other plots were planted with a susceptible hybrid called Macho. It's a plantain. And then we collected about 30 to 100 isolates per plot during 2 years. And we compare these samples with an inoculation method under controlled condition on leaf fragments we have developed in my team. And we were able, using this cross-inoculation experiment, to show that the isolates coming from the resistance hybrids were more, the blue, the one in blue here, were more significantly more pathogenic on these hybrids in comparison with the susceptible control. And there was no difference between the 2 samples, the 2 types of samples in the susceptible cultivars. So this was first evidence that the pathogen population adapt to the resistance hybrids. And to elucidate the genetic support of this adaptation, we use a population genomic approach, a genome scan approach. In this approach, we compare the samples coming from the susceptible and the resistant plots with SNP markers distributed along the genome with whole sequencing of full individual. In order to detect along the genome, to detect differentiated regions. I mean regions where the allelic frequencies between the two types of population is different in comparison with the other part of the genome. And this will be an indication that these regions are under host selection. So using pool sequencing of the whole genome of all populations, we are able to detect 32 genomic regions in total, which were putatively under host selection. And with the isolates from one population and from the individual sequencing of these isolates, we found in most of the regions two different haplotypes called here H2 and H1. And we then inoculate again the isolates having these two different haplotypes on both cultivar. And we found that the isolates, having the H1, the haplotype we call H1, were more pathogenic on the resistant host. As you can see here, you have here the level of the disease, and you see that the distribution for the isolates coming from the resistant cultivar is significantly higher. So we found this in 9 regions. By contrast, we also observe that isolates having the second one haplotype called H2 were actually advantageous, so more pathogenic on the susceptible host in 5 regions. So this experiment first confirmed that some of the regions we have detected are actually involved in host adaptation and pathogenicity. We found a polygenic adaptation architecture on both hosts with more regions involved on the adaptation on the resistant host. And in these genomic regions, we detect Candida genes. And this represents a starting point for further genomic studies of interaction to elucidate the role and the underlying mechanisms of these genes. So these results raise the question about the durability of resistance. And we can see here that in the case of banana and deficiencies, even it can take more time than qualitative resistance, quantitative resistance, such we have in the FIA18 hybrids, can be progressively eroded. And we think that combining different resistance mechanisms, it will be possible to limit pathogen adaptation if there are trade-offs between traits or antagonistic interaction. The pathogen population could evolve and the disease level of resistant plant could increase, but could stay below the one detected on the susceptible plants and stable during time. And this disease level could be agronomically acceptable. So we will have a balance between the efficiency of the resistance and the durability of the resistance. And it will be possible to combine different resistance and different mechanisms in the breeding program and we have developed at CIRAD. as Nabila presented, it's a reconstructive breeding scheme. So from a pool of diploid genitor, which can be improved in a pre-breeding phase, some of them are doubled with colchicine, and crossing diploid and tetraploid, we obtain on one cross triploid hybrids. And following this scheme, we were able to create new hybrids and disease resistance evaluation under control condition or in the field. We were able to demonstrate that these hybrids are both resistant to black leaf streak disease with a quantitative resistance, but also to Fusarium TR4. And also these hybrids present a new quality characteristic, and it could be useful to introduce diversification in the market and value chain multi-seat test with producer in Latin America and Caribbean are starting now in the framework of World Musa Alliance. It's a consortium we are building. Upstream to this scheme, so we are conducting some research. First, we study the structural genomic of banana, as Nabila presented, in order to orient and the breeding process, maybe in the pre-breeding phase. We also conduct a study to elucidate the genetic support and also functional genomics, so to elucidate the genetic support of traits, including host pathogen interaction, in the objective to help the selection during the process. So it will be also interesting to study pathogen population dynamic and adaptation to resistant genitor. So we are doing that on about 6 genitor we are using in the breeding, in our breeding program. These genitor are cultivars. First, we are evaluating the quantitative traits of interaction between Figensis and these genitors to see if there are different mechanisms. Then we will compare the adaptive architecture of Figensis population facing these resistant genitors to see if there are differences in adaptation speed. And finally, we are characterizing the genotype to genotype interactions between Figensis and the genitor to have an idea how to combine mechanisms to limit adaptation and exploiting a trade-off between traits. Yes, to finish my presentation, I would like to present you this preliminary results on the global genomic population analysis of Figensis. So we have sequenced about 96 Figensis isolates from different parts of the world, and we construct from this data this network to see the genetic divergence between these isolates. So you can see again that there is much more diversity in Asia. As you can see, the isolates from Africa or from Latin America or from Pacific are grouped together, and the diversity is lower in comparison with isolates coming from Asia, from the Philippines, from the continents of Asia, from Papua New Guinea, and from Indonesia, in this color. And also, more precisely, we detect a higher level of diversity in these regions, Indonesia and Papua New Guinea, because you see here, the isolates coming from this region, here, and also here, are distributed in the whole network. And also one isolate from Papua New Guinea, it's very divergent to all the other isolates. So this means that we did not cover the diversity existing in this region. Certainly, there are different populations which are very with a lot of differentiation. So these regions could correspond to an host pathogen coevolution zone. So we could have high diversity of host pathogen interaction in these regions, and it could represent first banana resistant source, and also a way to better understand the host pathogen interaction and the evolution of this interaction. And the first steps could be to describe the distribution and the population structure of the different fungal species, not only Figensis, but the two others, because they are also present in these regions, in parallel with diversity study of Acuminata subspecies. And so we are interested to develop such a collaboration with Indonesian researchers. To conclude, I would say that Sigatoka disease and meantime Fusarium disease can be controlled sustainably while reducing the use of fungicide by introducing diversity in cropping system at two different levels, at the interspecific level, with the introduction of hedgerow or association with other crops, and at the intraspecific level with the introduction of different resistances or cultivars. And diversification of cultivars, maybe not in Indonesia, but in other countries like in Africa and Latin America, when only one genotype is cultivated. So this diversification will also create added value for all players in the banana value chain. But research is still needed to better understand the underlying biological mechanisms and to define the spatial and temporal scales at which the diversification should take place. So this last slide is to present you the different people which were involved in this work, and at the bottom the sponsors. Thanks for your attention, and happy to answer to your question.
Thank you, Young. A very impressive presentation about one of the important disease in banana. So we are people from Indonesia. We should not only proud of to the center of diversity of banana, but we also should proud to the center of the diversity of the disease itself. Okay. So before I start the discussion session, I have a question. So this is a question from the organizer for Nabila and Young. So is it possible to have the PDF of your presentations to be shared to the attendee of this seminar?
Yes, sure. It's possible for me. Yeah, sure, no problem.
Okay, thank you, Young. Okay, so yeah, so you can send to me, and then I can send to the organizer. So anyone who is interested in the presentation, okay, you can contact the organizer then to get the PDF.
But I saw also that the presentation were recorded, so you should have also the presentation and the sound in the record, no?
Yes, and also the record is also possible to be shared in the YouTube.
It's no problem to share with you the PDF, no problem.
Okay, thank you very much. Okay, so now the discussion session. Yeah, actually, we already have one question from the chat previously during the Nabila sessions. So probably you can answer this question, Young. So this is question from Novianto here.
About plant resistance disease genetic, for the moment we have only few results. So only few QTLs has been published for Figensis, and also I think for Fusarium, at least for Figensis, and to be able to make a correlation between this genetic support and seasonal climatic or irrigation regime. we would have to compare plants or progeny in different situations. But for the moment, we don't have such data. So I think we don't have any idea about how plant disease resistance will be affected by seasonal climatic and irrigation.
Okay, thank you.
We need more study to that. First, we need to have a better idea on the genetic support, and then maybe to compare the expression of the genes we will have identified in different situations, or using condition under control condition, but we have to be able to contrast the environment to see if a different environment have an effect on the gene expression.
Okay, thank you, Young. Okay, so Mr. Nurfianto, is it okay? Are you still there? Okay, so I assume it's okay. Okay, so next questions. Anyone else? Okay, so there is a question in the chat box from, yes, still from the same person, from Nurfianto. Is there any relation between use of fungicide with soil health, especially to combat fusarium in banana?
I don't think so, but I don't think this has been studied yet. You know, because the leaf spot disease are leaf pathogen, so to control with fungicide, we spread fungicide on leaves, and after you have to test if the fungicide which go down on the floor has an effect on fusarium, but this has not been studied. I don't have one. I think certainly my guess is that this will have no effect on fusarium. If yes, no, I'm sure this will have no effect.
Okay, thank you, Young. Okay, thank you very much from Nurfianto in chat. Okay, next questions. Anyone else? If no, I have one question to you, Young. So we know that the Sigatoka is one of the fungus that do mating, right? But they need two different mating population, isn't it?
Mating population?
Yes. So is there any knowledge of the change because of the mating? So it must be there is a segregation or something in usually there must be a segregation on the progenies. Is there any knowledge about the rise of resistance fungus to the pesticides?
You mean about the genetic support of the resistant fungicide?
Yes, something like that.
Yes. What I can say about that, usually it's not using crosses, but we determine the genetic support of resistance and efficiencies is rather by homology with other system, because we use more or less the same molecule, for example, on wheat and on banana. And on wheat, with Zymocertoria tritici, which is a pathogen related to Pseudocercospora figensis. It has been demonstrated that one gene called CYP51, it's the gene involved, and the alleles in these genes are involved in the resistance to triazole. And so it's the same for figensis. And we found these genes in figensis. And also we found that isolates, some isolates having some alleles are actually resistant to triazole. It's the same for strobilurin, for example. We know the genes involved in the resistance, and we know exactly the mutation in our system. And we found the same in figensis. So it's the same genes involved in the resistance in strobilurin. And we found also that some of isolates having some mutation become resistant to strobilurin. So it's rather a comparison between isolates, a genetic comparison between isolates, and a correlation with susceptible or the resistance on the fungicide. Is not using crosses, but it's possible to cross actually two isolates in vitro. It's not easy, but it's possible to cross in vitro two isolates of figensis. And we are using that to try to identify QTL involved in pathogenicity. So it's another way than population genetic or population genomic analysis to elucidate the genetic support of pathogenicity. And we also develop studies using this approach.
Okay, thank you, Yang. Yes, thank you for the explanation. Okay, let's see. There is still no question in the chat box. Probably someone. And I think the question is not only for Yang. If there are any question for Nabila, I think it is so possible.
She's here.
Yes, I'm here.
Before we, yeah, now it's 37 past 2. Yeah, actually, normally it's 1.5 hour meetings, but maximum it can be 2 hours. I still give a chance to you if there are any questions. Otherwise, it is also possible if you have any question, something to do with the presentation by Yang and Nabila, you can send an email to me, or you can send email the organizer, and then organizer will send email to me, and I will deliver the question to Nabila and Yang. Okay?
Sure.
Yeah.
Sure.
Okay.
With pleasure.
Okay, I think this is the end of the Friday sharing because there is no more questions. The time and floor I return to the organizer. Thank you, everyone. Thank you, Yang, and also Nabila.